This lecture presents the Bioinformatics and Computational Systems Cascade, a structured computational framework developed under the research direction of Bryan Geniuson, research scientist at Natural Scientifics. The purpose of this presentation is to examine how multi-omics integration, network modeling, pharmacogenomics, and translational scoring can be unified into a coherent systems architecture appropriate for academic, clinical, and translational research environments. This presentation is educational and scientific in nature and does not constitute medical advice. Modern biomedical science produces enormous quantities of data: genomic sequencing datasets, transcriptomic expression matrices, proteomic abundance profiles, metabolomic flux analyses, large-scale clinical registries. However, scale does not ensure integration. Genomics is frequently analyzed without metabolic context. Network modeling may lack pharmacogenomic stratification. Clinical associations are often disconnected from mechanistic architecture. The Bioinformatics and Computational Systems Cascade addresses this fragmentation through hierarchical integration. Data acquisition leads into quality control. Quality control feeds multi-omics convergence. Multi-omics convergence informs network modeling. Network modeling integrates with variant mapping. Variant mapping supports translational scoring. Translational scoring produces structured clinical output. This structure is not merely sequential. It is regulated, feedback-aware, and evidence-tiered. The cascade begins with structured data ecosystems, chemical repositories, protein and pathway databases, genomic and transcriptomic archives, genome-wide association catalogs, clinical registries, internal compound analyses. Raw data is insufficient without integrity enforcement. Normalization pipelines prevent statistical distortion. Batch correction reduces systematic bias. Variant annotation ensures genomic consistency. Signal filtering eliminates spurious amplification. Reproducibility scoring quantifies methodological stability. Without these controls, computational modeling becomes visually impressive but scientifically unstable. Integrated biological layers include genomics, epigenomics, transcriptomics, proteomics, metabolomics, and microbiomics. The objective is concordance. Does genomic variation alter transcription? Does transcription modify protein abundance? Does protein modulation shift metabolic output? Does metabolic output correlate with disease burden? Multi-omics concordance scoring measures cross-layer reinforcement. When layers align, mechanistic certainty strengthens. When divergence appears, translational caution increases. Biology operates through interconnected systems: protein-protein interaction matrices, gene regulatory networks, metabolic flux systems, signal transduction cascades. Centrality analysis identifies hub nodes that may represent therapeutic leverage points. However, hub perturbation also carries toxicity risk. Systems modeling transforms isolated biomarkers into structured biological architecture. Mechanistic interpretation must be genotype-aware. Single nucleotide polymorphisms alter enzyme kinetics. Transporter variants modify absorption and clearance. HLA alleles influence immune response. ADME gene variation reshapes pharmacokinetics. The Variant Sensitivity Index stratifies metabolic velocity, transport capacity, immune susceptibility, and population allele frequency. Without variant mapping, translational models remain incomplete. The translational scoring engine integrates multi-omics concordance, network centrality weight, clinical evidence tier, computational confidence metrics, and variant risk modulation. The output is a Translational Readiness Index. This index quantifies structured plausibility. It does not claim therapeutic efficacy. It indicates probabilistic readiness for further investigation. Structured outputs include therapeutic prioritization matrices, pathophysiological burden mapping, personalized risk modeling, biomarker panel development, grant-ready analytical reports, and industry-aligned research dossiers. All outputs are evaluated across 4 distinct domains: mechanistic certainty, preclinical evidence, clinical relevance, translational readiness. These domains are intentionally separated to preserve scientific integrity. Mechanistic strength does not imply clinical validation. Clinical correlation does not guarantee mechanistic clarity. Computational modeling remains probabilistic. Docking simulations do not confirm in vivo biological activity. Network inference does not replace randomized controlled trials. Multi-omics concordance does not guarantee therapeutic outcome. Bioinformatics refines hypothesis precision. It does not eliminate uncertainty. Upcoming lectures will expand this architecture into deeper network centrality modeling methodologies, Bayesian multi-omics integration frameworks, variant-driven pharmacokinetic simulations, AI-assisted translational prioritization, real-world registry integration strategies, computational toxicology risk modeling, and grant-aligned translational scoring methodologies. This is part of an evolving research series exploring systems biology, computational integration, and translational architecture at increasing levels of analytical depth. Additional videos and expanded content are currently in development. The Bioinformatics and Computational Systems Cascade represents a structured maturation of computational biology within a systems-level translational framework developed under the research direction of Bryan Geniuson, research scientist at Natural Scientifics. If this work aligns with your research interests, you are invited to subscribe for continued academic briefings and structured research expansions. More detailed lectures, advanced technical modules, and applied computational frameworks are coming soon. Full scientific literature references are available for download. Thank you for your attention.